Hi everyone. So this week we want to talk a little bit about how we grow bacteria and cultivate them in the laboratory. We also want to talk a little bit about molds and a little bit about viruses because the methods you would use for each of those organisms would be a little bit different. Before I get into that demonstration, I first just wanted to remind you of some background information that you should already know, but let's just review it anyway just to make sure everybody remembers. So bacteria divide by binary fission. In one bacterium, as it grows, after the first generation, one bacteria will divide to become two. After the second generation, if all being equal, two will divide to become four. And after the third generation, 4 will divide to become 8. So in this way, when microbes grow on a solid medium in the laboratory, if we start with one bacterium, it will eventually grow to a large enough population to form a collection of bacteria, and those collection of bacteria are what we call colonies. And so if we take a look at a plate, each one of those little round circles represents the outgrowth of a single bacterium. So we can say that a bacterial colony, all of the progeny came from a single bacterium. When we report that in the laboratory, we don't usually count them as bacteria, we count them as colonies, and we infer that the number of colonies represents the original number of bacteria that we began with. So let's take a look at what bacterial colonies and mold mycelia look like in real time. Remember, last week we took a sample from the sink in the culinary lab, right? And from that sink, we actually were able to grow out many different kinds of microbes. So those such as forming these colonies that look something like these shiny ones or these yellow ones, small and large. These represent bacteria growing. Now, I can't say why we can know that those are bacteria. I can just say that based on experience, we know they're probably bacteria because of the way they look, their morphology. Likewise, this one right here is a different bacterium. And notice how dramatically different it looks. It has a distinctly different colony morphology. It's rippled. It's not smooth and round and circular, but yet this spreading yellow colony is certainly a bacterium. In contrast to that, I want you to look at this one right here now. So notice that one. If I turn the plate a little bit, we can see that that one is protruding out from the surface. And more interesting than that, we can see that that one, if I focus the camera there, you can see that there's these little white balls even there. Well, this one, if you look close at that, what's growing there, it's a little bit fuzzy. And from what goes on at home, you probably already know that that could be mold, and indeed it is. And those little round balls are the sport packages we would refer to as, in this case, their morphology, because they're nice and round. If you looked under the microscope, we would call them sporangium. So on a plate we have colonies and we have mycelia growing both at the same time. And in the next step, I'm going to come back and I'm going to show you what would we do to be able to separate them from one another. So why would you want to do this? If you were in a food company and you wanted to know what microbe might be causing food spoilage or a food safety issue, or maybe you were checking a fermentation to make sure the correct microbe is there, you need to be able to isolate them in pure culture and characterize them. Okay, be back in a minute. For food microbiologists, there's a specific language that we use to speak about microbes when we're speaking to each other, or if we're in a food company and we're writing a report. And so when we're speaking about bacteria growing, generally when we're speaking about situations on solid growth medium, we speak about bacteria forming colonies. And remember, a single colony represents the outgrowth of a single bacterium. Likewise, we often use that same nomenclature for yeast. So yeast is a fungi, not a bacterium, but still as they grow, they form what look very similar to bacterial colonies, and they are often referred to as colonies. In contrast, in the language of mycologists, that is, microbiologists that study molds, they'll form a unit on a plate, but it's not called a colony, it's called a mycelium. And in contrast to all of those, virologists, microbiologists who study viruses, they usually speak in terms of plaque formation. So I've provided you with a video to watch so that you could learn about what is a plaque. But remember that a single plaque that forms also represents the outgrowth of a single viral particle. So a common way to isolate in pure culture yeast in molds and bacteria is to streak for isolation. And in this method, it's very straightforward. If we imagine that we had our Petri plate, real one sitting right here, and we had a tool, an inoculating loop. Okay, so my marker will represent the inoculating loop. We could grab a colony of bacteria and then streak it onto the medium, and that's what we're going to do. So if I first did this with the marker, if I were to have touched the colony, I could then transfer those bacteria over to the agar plate. And then I would streak, without crossing back into where I entered, I would streak across the surface of the plate. In this way, I'm taking the bacteria that I removed from this plate and I'm spreading them out over the surface of this plate. Remember that in a single colony, one colony may have more than 1 times 10 to the 10th bacteria. So if I take from here and I put here, I'm spreading out 10 to the 10th bacteria. I will never have an isolated colony because there's so many bacteria there. So what I do is I sterilize my loop, sterilize it again, and then I would come back to here and I would take the sterile loop and I would take some of those bacteria and touch the surface and drag them out into this other area, and then I would spread them over the surface again. So 10 to the 10th here, I don't know how many here, but many less because I only grabbed a few and pulled them over. Likewise, I would sterilize my loop again, come back, and go to this area, take a few, and spread them out again. So after the medium grows and the bacteria are allowed to grow to large quantities, in this area, it will be heavily grown with bacteria, but no isolated colonies. Likewise, usually this area is heavy, and at some point, we're going to start getting isolated colonies that grow, of which each one is the outgrowth of a single bacterium. And if we take that again, we have a pure culture. To see the finished product, we would see something like this. So the first streak, after sterilizing the inoculating loop, the second streak, and then we're already getting isolated colonies before we have the third streak. So let's see how that's done. So we have Bunsen burner here. Get that lit. Okay. Inoculating loop. I can sterilize this loop with that hot flame. So I put it in the flame. Once it glows orange, I know that it's hot enough that it's killed any microbes on that inoculating loop. I let it cool before I do anything else. Then I can go to my sample from the sink and imagine that I want to isolate one of those bacteria. So for this exercise, I'm going to pick a very easy one. Let's say that I want to isolate the bacteria from this one white colony right here. So I take my loop and I pick up the colony, right? You can't see it, it's too small. Bacteria are microscopic, right? But that doesn't mean they're not there. There's plenty of them there. Go to my agar plate. On the surface, streak back and forth. Don't go back to where I started. Then I would sterilize the loop again. Let it cool. I rotate the plate. So I rotate it in my hand. And you can even see in the reflection where I already streaked before. So I'm just going to go to that area again. One time, drag a few out, and then I'm going to streak across the surface of the plate. So you can see the reflection of where I touched the plate again. I'll sterilize the loop again. I need to let it cool. I'll rotate that plate. And now I'm going to come back here one more time, go into there, and I'm going to streak out. And notice I used the whole surface of the plate. I started, let me sterilize my loop and get it out of the way. Okay, so look at the plate. Here's where I streaked the first time in this area, second and third area. This plate had been done before, so it had already grown. You can notice first area, second area, and third area, there's so few bacteria, nothing actually grew in that area. But we have isolated colonies. Each colony represents the growth of a single bacterium. For yeast and molds, we can use pretty much the same methodology for growth on a solid medium. For bacteria, in addition to solid media, we also have, and yeast we can do the same, we can use liquid medium. So here's a test tube. The only difference in the nutrients between this one and this one is that this one contains agar, which doesn't help in growth, it just makes the medium solid so we can have colonies form. This one, there's no agar, so it stays liquid. And if we inoculate that with bacteria, then shake them up because they settled out, you can see they grow and they make the medium turbid. So in every milliliter of medium here, there are approximately 10 to the 9th bacteria growing. Okay, so we can cultivate microbes, bacteria, yeasts, and molds on semi-solid medium. In liquid medium, we can cultivate bacteria quite easily, and we can grow yeast quite easily because they go in the suspension. We normally don't grow molds in liquid media because those mycelia, those masses, they don't disperse. They stay together, so they don't go into solution very well. So most people studying molds will prefer to grow molds on solid media.